Hello, welcome. Come on in. Hi, everyone. Hi, everybody. Welcome. We're still a minute early, so we'll give everybody just another minute. Hope everybody's doing well. Okay, it looks like people are coming in. That's wonderful. If you would, just go ahead and type your first name for me in the chat so I can take attendance. Very good. Thank you.
If you want to change owner to Vicky, because you're the only one that can do it, you have to go into the
Okay.
Yeah, I tried. You have to go into the chat box and as a participant and change it to Vicky because it won't. I can't do it.
Okay.
I tried.
I'll give that a shot, Vicky, okay?
All right. Thank you. Do the participant.
Yeah, I see you in here and I've got you. It's not letting me do it. I'll see if I can figure that out, Vicky. Of course, now I know you as owner. Strange.
Yeah, but nobody else does.
That's right. No one else knows who this owner person is. We'll see what we can do. All right, very good, everybody. It looks like everybody's here. I've heard from a couple people who aren't able to join us today. So we'll go ahead and get started. If you haven't already, go ahead and type your first name into chat. And then I'll have a record that you were here. I see that just about everybody has taken lecture exam 1. If you haven't done your exam yet for today, you still have several hours to do it. If for some reason there's something going on and you need to get up with me and maybe reschedule it for later tonight or something, you can do that as well. But just about everybody has completed the exam. And I do want to apologize because normally I send out a reminder announcement on the Friday before we have an exam, but my life got taken away from me on Friday with all kinds of crazy stuff that happened. And so I just didn't get a chance. And that's why I didn't send out a reminder till Saturday. I do apologize for that, though. I do like to remind you when you have a lecture exam coming up. And as I said in the announcement, I do have to grade your exams by hand. There's something like 5 questions that I have to grade by hand. So you'll know your lecture score, your first lecture exam score, once I have that done. And I try to get those done fairly quickly within a couple of days. So just hang on and you'll get those grades back. So today we're going to be talking about our next laboratory subject, which is gram staining. We talked about simple staining the last time we were together, and we said that simple staining involves one stain that we apply to a smear on a slide. And there's another kind of staining called differential staining that uses more than one stain. And gram staining is the single best example of differential staining. Gram staining is the single most commonly used staining procedure in microbiology. And that's because gram staining helps us to differentiate bacteria into one of two groups. So whenever we have a microbe that we don't know what it is, we need to identify it. Perhaps it's a diagnostic sample. We do a gram stain on that microbe, and it allows us to put it into one of two groups, at least to start with. And that's what we call gram positive and gram negative. And we'll talk today about how we make that determination. Now, that's not where we finish when we're trying to identify a microbe. That's just the first step. But once we know that something is gram positive or gram negative, we already know a lot about it. So it helps us in planning what other diagnostic tests we want to do on that microbe in order to identify it. So this is a very, very commonly done test. Like I said, it's generally the very first test that's done on a microbe that we're trying to identify. And it's a differential staining procedure, not a hard one, but our first one that we'll talk about. So before I get started, I just want to ask if anybody has any questions about anything else that you've been doing. I finished grading your lab questions from last week, and those would have, I made some comments on some of your submissions if you missed questions. If you haven't had a chance to look at that yet, that's fine. But if anybody has any questions about those lab homework questions, we can certainly talk about that. Or if you have any questions about anything else that you've been looking at or learning about, we can certainly talk about those. I know
I
you're
have a question.
Yeah.
Sorry. So it was the clinical one, I believe. It was, so you can see bacteria, and what was it? Protozoa.
Oh, that's right. Yep.
Under the light microscope, but you have to be at a higher lens to see the bacteria.
That's right.
Is that correct? Okay.
That's exactly right. So the question read something like that: you're under the 4X lens. And remember, that's the low power. On a light microscope, depending on the brand of microscope, it's the low power lens is either going to be 4X or 5X, but very low power, obviously. We can't see bacteria under that low power lens. You have to go up to at least 40 to see them. And that it's even better to go up to the 100X oil immersion lens. Remember, we call the 40X objective lens, we call that the high dry lens, because we're at a pretty high magnification, but we don't use any oil with that one. And then the highest power lens we have is the 100X lens, which is the oil immersion lens. So in that question, what should have caught your eye, hopefully, was the 4X, that you're looking at a sample under a 4X lens, and you can see a microbe and you can see it moving. You can see that it's modal. And the question said something like, you decide that it's probably not a bacterium. I think you're told that it's a fecal sample too. So you decide that it's not a fecal bacterium, but probably a fecal protozoa that is mobile. So here's what you would have need to have determined in that clinical case. Number one, you can't see bacteria swimming around under that 4X lens, but you could see a protozoa under that low power lens. If you remember from the lecture called The Cell, protozoa are single-celled, microscopic organisms. So they're microbes, but they're not like bacteria and archaea. They are eukarya. They're like you and I, they're eukaryotes. And that means that their cells are bigger than bacterial cells. They're bigger because they have to be bigger, right? Eukaryotes, like humans and other organisms, have organelles. And so our cells are just bigger. So protozoa are single-celled organisms, but they're eukaryotic. They have organelles inside of them, so they're relatively large. And a fecal protozoa would be visible underneath a 4X lens. So remember, when you're taking those, when you're answering those clinical application questions on your homework, there's stuff to think about in those questions. Be sure to read it to yourself a couple of times. Make sure you understand what's being asked of you, number one. And number two, make sure you think about all the clues that you're given. So the big
Thank you.
Yep, no problem. Anything else? Good question. All right. I'm going to go ahead then and pull up our lab slides for today, and we'll start talking about this very common test procedure called gram staining. And as always, I'll stop periodically to ask if anybody has any questions, but you can also just jump in and ask if you need to before you forget your question or anything. Don't hesitate. It's fine. So gram staining got its name because of the person who discovered it and first described it. So the reason that you see this word gram spelled with a capital letter is because it's somebody's last name. So what we're going to be talking about today is a couple of things about this gram staining procedure. We're going to talk about why we do it, what the purpose of it is. We're going to talk about the role of each one of the stains in this procedure. We're going to walk through the procedure together. I've put together some images demonstrating how you do this procedure. And then we'll spend a little bit of time towards the end of class looking at some gram stained smears. And we'll see if we can figure out what the gram status is for those organisms. That's what we call the results. We call that gram status. And we'll finish up today talking just a little bit about some of the limitations of gram staining, because every test that we use diagnostically is going to have some limitations associated with it. So the purpose of gram staining is to take an unknown type of bacteria and assign it to one of two big groups, either what we call gram positive or gram negative. And this status is all about the kind of cell wall that is around that particular bacterium. This week in your lecture material, you're going to be learning about prokaryotic cells. And there are two kinds of prokaryotic organisms. There are bacteria and there are archaea. Those are both prokaryotes. And then the last group is the eukaryotes, like I mentioned a minute ago. So inside the prokaryote domain, the prokaryote classification, if we look into the bacteria, those organisms as a whole not only have a membrane around their cells, but they also have a cell wall around the membrane. And in bacteria, the cell wall is made out of a very unique molecule. We don't find this molecule anywhere else in nature. And that molecule is called peptidoglycan. It's just a type of carbohydrate, but it's a very particular, very unusual carbohydrate that gives that wall a lot of strength. And it helps protect the bacterial cell out in nature. Now, when an organism is called gram positive, what that means is that the peptidoglycan that makes up their cell wall is very, very thick. When an organism is called gram negative, it means that the peptidoglycan in their cell wall is very, very thin. Now, gram negative organisms, the ones that have the thin layer of peptidoglycan, also have a second membrane. So they have a membrane around the cell itself. All cells have a membrane, remember. Then there's a cell wall made out of peptidoglycan. And then there's a second outer membrane in these particular types of bacteria. Very, very interesting, weird structure. So the gram staining procedure is going to tell us whether a bacterium is gram positive or gram negative. And there is another category that's called gram indeterminate. There are a particular type of organism that will not gram stain, a particular type of bacteria that we'll talk about today, that just don't gram stain. They don't take it up. They don't take up any of the color from these stains. Most organisms, though, fall into either gram positive or gram negative if they're bacteria. We call the results of gram staining, we say that that is the organism's gram status. And when we use that phrase, when we say, oh, the gram status of this bacterium, what we're going to report is whether they're gram positive or negative and what shape the cell is. So for example, I might say, well, the gram status of Staphylococcus aureus is a gram positive coccus. Or I might say the gram status of E. coli is a gram negative rod. I'm going to report whether it's gram positive or gram negative, and I'm going to report the shape of the cell when I report a gram status. And like I said earlier, the nice thing about gram staining is it's really the first of many tests that we have available to us when we want to determine the identity of a bacterium. Now, if you take a look at the pictures on this slide, what you're looking at on the left represents a gram negative bacterium, and on the right, a gram positive. Now, this is not the entire bacterial cell here. What the artist has done is shown us just a little piece of the membrane that surrounds a bacterial cell, just a little straight piece of it. And that's what's brown here down at the bottom of the slide. You can see, we know that this is membrane because you can see the heads and the tails in the membrane. So that represents the cell membrane, or what we call the plasma membrane. This artist has it labeled as the cytoplasmic membrane. All those terms mean the same thing. Now, remember, when you go inside of a cell, that's where you find all the cytoplasm, and that's where you would find the DNA and everything else. That would be down at the bottom of the slide where the white area is. But if we go towards the outside of the cell, that's where we're going to hit the cell wall and any other structures that might be out there. So in this drawing, the purple represents the cell wall, and it's made out of peptidoglycan, as you can see with the label. So the gram-negative bacterium has a very thin layer of peptidoglycan outside of its cell membrane. Next door to it, you can see lots of purple, and that represents a gram-positive organism. The gram-positive bacterium still has the brown cell membrane around it. And then if we go outside of that membrane, you can see this thick layer of this purple peptidoglycan. Now, it's important to note that the gram-negative cell also has a second membrane. It has what we call an outer membrane. And that membrane is literally attached to the peptidoglycan layer. If you think about it, it makes sense. If the cell wall is there to protect this cell, remember, bacteria are single-celled organisms. Their whole organism is one cell. So if the cell wall is there to keep that one cell as safe as possible, it makes sense that organisms that have a little thin layer of peptidoglycan have something else as well. They have a thin layer of peptidoglycan, and they also have a second outer membrane. And those two things together will help them stay alive out in the world, out in the environment. Now,
Dr.
grams,
G,
yes,
sorry.
that's
At
okay.
the other picture, what was the green above the outer membrane? You might have said it.
Oh,
and I
yeah,
missed
yeah.
it.
Okay. I actually didn't mention it, but we can, I'll briefly mention that now. What's drawn out here in green is a molecule called lipopolysaccharide, or what we shorthand as LPS. Now, this molecule that you find on the very outer surface of a gram-negative bacterial cell is there to help protect the cell, but it's actually an important molecule in medicine. We call this LPS chemical, we give it another name too. We call it endotoxin. And there are times in humans when, and I don't want to go too far into it right now, but there are times in humans when they get certain medical conditions where a lot of the gram-negative bacteria inside of our intestines will die. They all die at the same time. And what happens is all those dead gram-negative cells, they fall apart, they start to degrade. And this green LPS molecule gets released and absorbed into our bloodstream. And it's actually quite toxic to animals, humans and other animals. So we give it a name, we call it endotoxin. It doesn't bother us when the gram-negative bacterium is alive and swimming around in our intestine. It doesn't bother us at all. But if that cell dies, especially if lots of those cells die off together because the person is sick, that green molecule can actually make humans and other animals very, very sick. You'll be learning more about that green molecule as we move ahead in the course. Good question. So gram staining is a differential type of staining because it involves different stains. We're not going to just apply one stain and then rinse it off. We're going to apply one stain, we're going to rinse that off, we're going to apply another stain and rinse that off, and so on. We also consider gram staining to be a direct staining procedure. Now, there is a particular and an important step in gram staining called decolorizing. In fact, gram staining depends on this decolorizing step. Like the name suggests, we're going to apply a chemical that's going to remove some of the stain that we've already put on. That may seem silly, like why would you bother putting stain on and then taking it off? But it will make sense to you in just a minute. We're going to decolorize as part of this procedure, and we're going to do something called counter staining. Now, the chemicals, the stains and other chemicals in the order that we apply them are called crystal violet. I showed you some cells that were stained with crystal violet when we were together last time. Crystal violet is a primary stain. It's a stain, in other words, that gets used by itself all the time. And it's the primary stain in this protocol. It's the first and critical stain in this protocol. It gives the cells a purpley blue color. So we put the crystal violet on and then we rinse it off. Then we put on a chemical that's called Graham's iodine. This is basic iodine with another chemical that's been added to it. And what Graham's iodine does in this procedure is it acts as something that we call a mordant. That word is right here on the slide for you. If you've ever had any experience at all dyeing fiber, dyeing yarn, dyeing t-shirts, anything like that, when we dye material and we want the dye to adhere to the material, you have to add a chemical called a mordant. So in Graham staining, Graham's iodine is going to help the crystal violet penetrate into the peptidoglycan and stick to it. Let me say that again, because that's an important point. The Graham's iodine, the mordant, is going to help the crystal violet penetrate the peptidoglycan in the cell wall and stick to it. Now, the third step is the decolorizer step. Decolorizer is just an alcohol chemical.
Is there another word for pepto? Is it a protein?
Peptidoglycan. No, I'm afraid that's the only name it has, and it's a horrible word to spell.
So pepto.
It's peptidoglycan.
And you said that
Here
the
it is.
mordant
Here.
helps. Oh, you said the mordant.
Yeah, go ahead. The mordant helps.
I spelled it right. Surprise.
The mordant
helps
the
crystal violet.
Yeah, it's going to help it penetrate into this peptidoglycan and stick to it or adhere to it.
Okay, thank you. Sorry.
Yep, no, that's okay. So we're going to apply the decolorizer in the third step, and we're going to do it very carefully. We're going to do it for a very limited amount of time. And what the decolorizer is going to do is take away some of the crystal violet. It's not going to take away all of it, but we'll apply it long enough so that it takes away some of it. And again, this will make more sense in just a minute. The final step of the process involves what we call a counter stain. Since we're taking away some crystal violet with the decolorizer, we need to add back another stain. And the other stain we add back is called safranin. Now, you saw some cells that were stained with safranin last time. Safranin gives cells this lovely pinkish red color. So take a look at this slide. And this slide might make some of this make a little more sense. What you're looking at on this slide is an organism over on the left that's gram positive. It also happens to be a coccus. And an organism over on the right that's gram negative and happens to be a rod shape. And we're going to go from top to bottom through the gram stain procedure with the colors here so we can see what's happening with the colors. So remember, the first thing we do is we make a smear and we fixate the smear. We either do a chemical fix or we do a heat fixation. And then we're going to apply our first stain, which is crystal violet. Crystal violet likes to stick to peptidoglycan and some other materials. So both the gram positive cell and the gram negative cell will turn purple when they get exposed to crystal violet. Remember, the next step is to apply Graham's iodine. And we said that's going to make the crystal violet penetrate the peptidoglycan and stick to it. So they're showing us a slightly different color of purple here just to represent that it's really penetrating through that material. Now we're going to decolorize. We're going to put a little bit of that alcohol solution on top of the cells. And some of the crystal violet is going to come out of the peptidoglycan, but not all of it. Only the outermost layer is going to lose crystal violet. So if you look at the gram-positive cell, the gram-positive cell is going to stay purple. And the reason it stays purple is because it has lots of peptidoglycan. It's got a big, thick layer, several different layers of that peptidoglycan chemical, that material. The gram-negative cell is different. The gram-negative cell has a very thin layer of peptidoglycan, and the decolorizer is going to pull all the purple out of it. The gram-positive cell loses some purple with the decolorizer, but there's plenty of purple left behind because there's such a thick, thick layer of peptidoglycan. The gram-negative cell is going to lose its purple because it doesn't have much peptidoglycan to start with. So when we decolorize it, we basically take it away. Now the last step is to counterstain with safranin. The gram-positive cell can't take up the safranin because it's already got the crystal violet in it for the most part. So the gram-positive cell stays purple. The gram-negative cell lost all of its purple, so it will take up the safranin and it will stain pink. So this is a 4-step procedure: crystal violet, then Graham's iodine, then decolorizing, then counterstaining with safranin.
How long does this take?
It doesn't take long at all because, as you'll see in just a minute when we walk through it together, we're only leaving these stains on the slide for somewhere between 30 and 60 seconds each. The hard part about Graham staining is that you got to rinse off stains in between. And the hardest part is the decolorizing, because we're going to limit it to about 10 seconds. We're going to decolorize for about 10 seconds, and then we're going to stop. So on this slide, what you're looking at is just a list of what I said for this previous slide. It's just written out in words, what I just said a minute ago. So you're going to make your smear, you're going to apply your crystal violet, then you're going to rinse that off. You're going to apply the Graham's iodine, then you're going to rinse that off. You're going to apply the decolorizer for 10 seconds only, and you're going to apply it drop by drop. I'm going to show you how to do that. And then you're going to rinse that off. Then you're going to put your safranin counterstain on. Then you're going to rinse that off, and then you're going to blot your slide dry. So 4 steps with rinses in between them. Now it's important to note that when we rinse off a slide that we're Graham staining, or frankly, we're putting any kind of stain on, we want to use distilled water as our rinser. Now, why do you think we want to do that? Why would it be a bad idea for us to use, say, tap water to rinse off our slides? Any ideas?
You don't want organisms that are already in the water,
Yes.
in the tap water.
We don't like to think about that, but there can be a small number of microbes in our tap water. So there's that problem. But the other issue we have to think about is any chemicals that might be in our tap water that might disturb our stain. Okay, so you just have to be aware of the fact that you want a nice pure water, essentially, when you're rinsing. So you don't add anything you don't mean to your slide. All right. So let's walk through this with some pictures now so you can see how you would do this in the laboratory. So in this first slide, you can see I'm holding a glass test tube that has some culture in it. So I put some broth in the bottom of this tube, what we would call a nutrient broth that has all the little nutrients in it that bacteria like to grow. And I put this tube, I inoculated this tube with a pure bacterial culture. And then I put it back in the incubator and I let the bacterial cells grow and divide. Now I'm going to gram stain a sample of this. So you can see my inoculating loop here. I would have taken that loop and I would have sterilized it first by putting it in the incinerator, as we talked about last time. You'll notice that once I have this tube open, I'm keeping it at an angle in my gloved hand. I'm holding the lid in between my fingers so it doesn't get contaminated by putting it down. And I'm putting that inoculating loop in right around to the middle of the culture volume after I gave it a good mix. Now, this is just a very short video clip, but you'll see that I've taken my loop here that has a little bit of culture in it, and I'm just basically making circular motions on my slide. I'm spreading out that small amount of liquid culture. Remember, you're only going to fill that little tiny loop space with culture, but it's enough. It's enough to spread out in the middle third of your glass slide and create a smear. Remember, all a smear is a thin layer of cells. We don't want a thick layer. It might be too hard for us to see. So we're going to make a nice thin layer of cells by spreading out that material in the middle third of the slide. Now, we talked about the importance of letting slides dry. You can certainly just put a slide down and let it dry. In this short video, you can see that I'm just waving the slide up and down. That's fine. See how I'm holding the slide at an angle? And I'm just waving it back and forth. That can help it dry a little bit quicker. Sometimes I think that just makes us feel like we're doing something to help it dry. I'm not sure if it does a lot.
Is that always safe to do? Like, what if we're using bacteria that is like toxic to us?
So remember that if you were working with a pathogen, like let's say you were in a diagnostic lab and you were working with a sample that might have a pathogenic microbe in it, you would either put this under one of those biological hoods while you were shaking it like this, or you wouldn't do this. Anytime you're handling something that might be pathogenic, you just want to be extra, extra careful with it. Now, remember though, bacteria cannot jump up. So if there's fluid on this slide that has microbes in it, my shaking it back and forth like this is not going to let them jump off the slide. So it's not nearly as dangerous to do that as it would be to heat this thing up when it was wet. We talked about how heating can aerosolize the cells. That's very dangerous. So some people like to shake like this. Other people like to just leave the slide on the desktop. And remember, you also have the option of using that slide warmer that I showed you last week, the one that looks like a hot plate. You can set your slides on that warmer and let them dry that way as well. Now, once the slide is completely dry, I'm going to heat fix it. So I've plugged in my incinerator. I let it get nice and hot. I'm going to get my clothespin and attach my slide to it and just gently rest the smeared area on top of the metal. And you do want to touch the slide to the metal. The metal doesn't get nearly as hot as the inside tube does, but it'll get hot enough to allow you to fix this slide. And remember, you want to let the slide sit on that metal housing for about 20 seconds. Now on this picture, you can see 3 different slides. I've got them all labeled. You can see that I've taken a Sharpie pen and I've drawn a circle in the middle of each one of these slides. When you're first learning how to do smears and how to stain and how to focus a microscope and things like that, it really is helpful to use a marker this way. It's just going to help you find your smear when you go underneath the microscope, when you get to that point. People who are more experienced making smears don't do this. They can skip that part because they're very good. They're very experienced at finding cells under the microscope. The other thing I put this picture in for is to remind us that if we have several slides to examine, let's say one morning in the laboratory, we often will gram stain all of them at the same time. Now, once the smear is dry and we've done our heat fixation step, now we're ready to do the staining procedure. What you can see on this slide is just a very simple, low-tech, what we call a staining tray. And there's all kinds of versions of this in microbiology laboratories. This one is made out of just a basic, inexpensive Tupperware plastic dish. And all we've done is we cut some little holes into the plastic on each side, and we strung some zip ties across that plastic box. So this is just the solid, plastic zip ties that you can buy at a hardware store. And what you do when you do that is you create a little ledge that sits above this staining tray. Now I've got 1, 2, 3 glass slides that I have laid down on top of those zip ties. So they are suspended over this Tupperware box. Now, the reason that we use a staining tray is because stains are not suitable for throwing away down the sink. We cannot put them down the drain because they're dangerous chemicals. They're considered hazardous. So anytime we do staining in the laboratory, we have to do it over a container that can catch it. We need to catch all of the stain, all of that rinse water, all of the decolorizer that we use, all of the iodine that we use. We want to catch it. So we catch it in this little staining tray. And then when we're finished staining at the end of the day, we would empty this tray into a waste bottle. And, a couple times a year, depending on how busy the lab is, a couple times a year, you would call for a hazardous waste pickup, and you would send that stain off to be appropriately disposed of. Now, gram staining is so common that you can go to any chemical supply house, any chemical supply catalog or website, and you can buy a kit for gram staining, and that's what this is. This one happens to come from a supply house called Becton Dickinson. And what they sell are these 1-liter bottles of each one of the chemicals for gram staining. So depending again on how busy your lab is, you can do many, 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 many gram stains with this big bottle of each one of these chemicals. Some microbiology labs go through one of these kits a week. And some microbiology labs won't even go through this in a year. It just depends on how busy your lab is. So on this slide, you can see our staining tray right here. I've got our 3 slides sitting on top of the plastic zip ties. So there's a space between the slide and the bottom of the staining tray. And you can see that I'm using a pipetter to place the first stain over my smears. And remember, that first stain is called crystal violet. Remember, these stains are not hugely expensive. And so we are not stingy with them when we do our staining. I happen to have a circle drawn around my smears, but I want you to remember that you want to flood your smear with crystal violet. You want to ensure that your entire smear is underneath crystal violet, because you want to make sure you stain the cells. The last thing you would want to do is put on too little crystal violet and only have, a quarter of your cells get stained. We're going to put the crystal violet on in a puddle, basically, and then we're going to let it sit for 30 seconds. Just 30 seconds is fine. Now, that 30-second time period is not strict. And what I mean by that is if you put it on for 25 seconds or 32 seconds, it's not going to matter. About 30 seconds, though, is what you want to go for. Now I've picked my slide back up here with my clothespin, and I've got myself a nice clean dropper, and I'm going to rinse off my crystal violet with my distilled water. I'm just going to squirt my distilled water on top of the smeared area. Notice that I'm holding the slide down at an angle so that all of the crystal violet, all of the rinse water is going to go down into my stain tray. So this is what the slide looks like after I rinsed off the crystal violet. And I want you to notice that you can't see anything, right? You don't see a smear here. You don't see a big smear of purple. You just see a clear area. And that's because a well-made smear is a very thin layer of cells. You're staining the cells. You're not staining the background. So it's perfectly normal not to see anything. Now we're ready for the Graham's iodine. Remember, this is our mordant. And the job of a mordant is to help a stain adhere and penetrate a material. We use mordants when we dye yarn. We use them when we dye clothing, the materials that we use for clothing. Anytime you're going to dye a material, you're going to add a mordant to help that dye really penetrate and adhere. Remember, Graham's iodine is not regular iodine. It's not the kind of iodine that you would buy, say, at a hardware store or a chemical supply store. It has an extra ingredient added into it. So here are my slides back on my twist ties, back on my slide tray. And now they have a big puddle of Graham's iodine sitting over them. Now we're going to leave the Graham's iodine for, again, about 30 seconds. And that should be plenty long enough. I'm going to rinse off my Graham's iodine now. Again, I'm tilting my slide so all of that chemical goes into my stain tray. Now we're at the decolorizer step, and this is the step that drives students crazy because it is possible to mess this up. The way you would mess up this step is by leaving the decolorizer on for too long. We're not going to put a puddle of decolorizer on our slides. We're going to drip it down the slide. If we were to set a puddle of decolorizer on top of these smears, and we were to leave it for, say, a minute, it would pull all of the crystal violet out from both gram-positive and gram-negative cells. So you can overdo this step. You can over decolorize. So you have to learn how to do it correctly. And that's what I'm showing you in this picture here. So I'm holding the slide again with my clothespin at an angle over the stain tray. I've got a nice fresh pipette here. I've pulled up some of the decolorizer. And while I'm counting to 10 in my head, I'm going to drip the decolorizer. See what I'm doing there? Drip, drip, drip over the smear. You want to make sure that you hit every area of the smear, but you want to do it for 10 seconds and then stop. Here, let me play it again for you. Drip, drip, drip down that slide. And if you do it for 10 seconds and you get the whole smear covered, what that's going to do is pull the purple dye, the crystal violet, out of the gram-negative cells while leaving plenty behind in the gram-positive cells. Now, very important, you have to wash decolorizer off, just like you wash all the other stains off. Remember, I said if you leave decolorizer on too long, it will pull the crystal violet even out of a thick layer of peptidoglycan. So after you do your drip, drip, drip decolorizer, you just take your distilled water and you rinse it off. It's easy to forget to do that because decolorizer is clear. It's just an alcohol, so it's clear, but it's an important step to remember. Now we're ready for the last step, which is the counter stain. You can see I overdid it here with the saffron in. I ended up putting saffron in over the entire slide, not just the smear. But the smear, again, is right here in the middle. I wanted to make sure I had enough saffron in to cover the smear. Now, the saffron in needs a little extra time. So we leave saffron in on for a whole minute. Again, approximately. We leave the crystal violet on for 30 seconds. We leave the grams iodine on for 30 seconds. But we're going to put the saffron in on for a full minute. When we're finished with that, we simply rinse it off. This is that special paper I told you about that we can use to blot our slides dry so we can see how we did. So here are the 3 slides that I just gram stained. I want you to notice a couple things. Number one, there are some blotchy areas on these slides that are purpley and blue and maybe a little pink. That's okay. Don't worry about that. It's perfectly normal on a slide. The other thing I want you to notice is that some of the Sharpie marker is gone. Can you see that? Some of the Sharpie has washed away. The decolorizer will take Sharpie off. So not everybody likes using Sharpie pens in a microbiology lab. There's a couple ways around this though. The first thing you could do is choose a different kind of pen. And instead of using a Sharpie pen, you could use what's called a grease pencil. You may have seen these in some of your classes. This pencil literally has like a wax material in it. If you draw a circle with a grease pencil, it won't come off in the decolorizer. The other trick that some people use is they don't put the Sharpie on the same side as the smear. Instead, they draw their circle on the bottom of the slide and they put their smear on the top. So all the stains and all the decolorizer will be applied to the top and the circle on the bottom will stay untouched. Like I said a few moments ago, most experienced laboratory technicians don't draw a circle. But while you're learning, it is a good trick to help you find your smear. All right, before we look at some results, does anybody have any questions?
Yes.
Go ahead.
So there's like a total of 7 steps.
Right, with the rinses, right?
Yeah. Because flood, rinse, iodine, rinse, Decolorizer.
decolorizer, rinse,
Rinse.
and then
So it's
soften, and then rinse.
then, oh, rinse. I didn't put, so 8.
Yeah, you're going to want to rinse that last stain off too. And then you can blot them dry. And remember, your slides don't have to be like bone dry for you to put them on a microscope and examine them. You just don't want to have them, soaking wet, covered in water. But if they've got a tiny amount of water on them, that's fine. You just want to blot them dry as best you can get them before you take a peek at them under the microscope.
I
All
have a
right.
question.
Yeah.
So for the gram decolorizer, the 10 seconds is as you're dripping it, right?
As you're dripping it. And some
And-
students will say to me, how fast should I drip it? And I always say to them, you just do it. Just push, push on your stopper, your bulb on your pipetter. Just drip the colorizer down, count to 10. And it will either you'll either have done it great or you will have pulled all the stain out. You probably can't pull the stain out in 10 seconds. So I always tell students, it doesn't really matter how fast you drip it. The important thing is that you don't go too long.
And then after the 10 seconds of dripping, you can put it right under the distilled water, or
Yeah,
you can
then you just rinse
okay.
it. Then you just rinse it.
Okay.
It's amazing and it's tough. Some people will tell you that when they decolorize, they can see some purple coming out. Like as the decolorizer slides off, they can see sort of a purpley sheen in it. I don't tend to see that when I do it, but I just know that if I do it for 10 seconds, chances are it'll be plenty. And then I'm ready to decolorize, but I mean to counter stain. But it's really important that you remember to rinse after you do that decolorizer because you got to get that alcohol off. Good question. All right, so let's take a look at some results now and see what we can figure out. So on this first slide, we're looking at a bacterial smear. We're at 1000x total magnification. So we're using the oil immersion 100x lens. And then remember, we also have to multiply that by 10 to account for the eyepieces. So our total magnification is 1,000 times. Now on this slide, I think you can see two very distinct colors. Yeah, you can see a purpley color and you can see a pink color. This is a great example of a slide that has both a gram-positive organism on it and a gram-negative organism. Now, what shape do you think that purple organism is?
Caucus.
Caucus, good. These are circular cells. See them? Now they're kind of clustered up together in places, so it's not immediately apparent, but if you look carefully, you can see that each one of these cells is actually a circle. So there's lots of cells and they're kind of clumped up together in areas. That is what we would call a gram-positive caucus. So its gram status is gram-positive caucus. Now this pink organism is different, isn't it? This one is rod-shaped. And again, the rods are clustered up in areas like it's hard to tell what shape they are in this little area here or this little area. But you can see some pairs of rods in places. You can even see a single rod, maybe right here. Here's a couple single rods right there. So this second organism has a gram status of gram-negative rod. You could also say gram-negative bacillus, but most people that I work with just say rod. It's a more common expression. Now, a lot of cocci that we deal with in the medical field, a lot of our gram-positive cocci are things like Staphylococcus and Streptococcus. So it's tempting sometimes to think that all cocci are gram positive, but they're not. There are gram negative cocci as well. And the same is true for rods. A lot of what we see in human medicine and in veterinary medicine, a lot of what we see is gram negative rods. So it's tempting to think that all rods are gram negative, but that's not true. There are some gram positive rods as well. Here's another mixed culture. What do you see? What colors do you see? Do you see purpley blue?
Yeah, and pink.
And pink, right? Two different organisms are on this slide. You can tell that both organisms are approximately the same size, and they both have the same shape, don't they? This is both a gram positive rod and a gram negative rod. So this is what we would consider a mixed culture or a mixed specimen. Now, on this slide, up at the top, you're looking at this gram stained smear under the 40x objective lens. So the cells look a lot smaller. Down here on the bottom, this one is under the 100x objective lens. So this one's a more magnified sample. So even under 40x, you can see these cells, right? You can see them. It's not as good a view as under 100x, like this slide, but you can still see bacteria under the 40x lens. So these are gram positive, and these are caucuses or cocci. Now, the reason I put this in here at 40x is I want you to get a sense of cells arranging themselves on a slide. This happens to be a Staphylococcus, and Staphylococcus likes to make these clusters. People call them grape-like clusters, like a bunch of grapes. There's one here, and there's one here. There's one here, and maybe one next to it. They're just clusters of the cells. In other words, these cells are not evenly distributed across the surface of that slide. They're purple, so they're gram positive, and they're round, so they're cocci. And one of the things we know about Staphylococci are that they tend to form these grape clusters on a microscope slide. Now, if you go down to the bottom, this is a gram negative rod. And again, this one's under a little higher power, so we can see it a little more clearly. This is a bacillus species. There are several important bacillus organisms in medicine. Bacillus can be an organism that is perfectly harmless, and it can also be a pathogen. So some bacillus organisms are dangerous. They cause disease. A good example of that would be Bacillus anthracis, which is the organism that causes anthrax. But there are other bacillus species that, for example, live in our gut, and they're important. They're an important part of our health. So not all of them are bad, just like not all Staphylococcus are bad. Usually, there are good and bad strains of bacteria. This is a nice slide. You've actually seen this slide before. What do you think? What would you call the gram status of this organism? Feel free to unmute yourself if you want to. Or you,
Positive caucus,
right, gram
gram
positive
positive caucus.
caucus. That's right. Now, you might be able to convince me that this organism is also clumping up in grape clusters. I can see a grape looking cluster right there. So who knows, it might be a staphylococcus, but its gram status is just whether it's positive or negative and what shape it is. So that's all we say with gram status is whether it's gram positive or gram negative and what the shape of the cell is. Now, take a look at this slide. We are under the oil immersion lens here, where therefore we're at 1,000 total magnification. This came out of a clinical sample, which is why the background of the slide has picked up a little bit of stain. We don't ordinarily see that. First of all, I want you to just look at the cell, each individual cell here and the shape, and tell me what you think the gram status is. So ask yourself, is it purple or pink? Are the cells purple or pink? If they're purple, it's gram positive. If they're pink, it's gram negative.
Gram positive.
Gram positive. Good. And this is another caucus. Good. I can see 2 cells next to each other right there. I can see 2 cells down here. I can see 4 of them right here. I want you to notice how they're arranged. These cells are not arranged in a grape-like cluster. These cells have arranged themselves into chains. Can you see that? They're all aligned one after the other in a long chain. This is a feature of Streptococcus. So Streptococcus likes to form these long twisting chains when it's on a slide. Now, on both of these images, the one on the left and the one on the right, we are under the high dry lens, the 40x lens. So again, a little bit harder to see, but we can still see. We can still see this. You can definitely see the color, right? If you needed to go up to a 100x lens to get more detail, you could do that. But just by seeing this dark color right here, I could automatically say, oh, this is gram positive. These happen to be gram positive cocci. Don't worry if you can't see individual cocci on here. I would expect that you would need to see them under the 100x lens to really tell. Over here, we've got a mix. This is a combination of both gram positive cocci and it's also gram negative rods. Again, they would be hard to see for you under 40x, but I think you can see the purple color and the pink color. Now, if you were growing a culture of bacteria in the laboratory, if you were purposefully growing a culture, that would be what we would call a pure culture. It should only have the organism that you're growing in it. But a lot of times when we get clinical samples, we're going to get mixed cultures. There's going to be more than one organism in there. Typically, though, if we have a patient who is suffering with a disease that was caused by the bacteria, one of the bacteria is going to predominate. In other words, sometimes a person who has pneumonia, if you get a sputum sample and you take it to the microbiology lab and you grow those cells in culture, it's not uncommon for you to find 2 or 3 different types of bacteria in there. But one of those types is going to grow the most. And that's usually the one that is the culprit in the pneumonia. You've actually seen the organism on this slide before. What do you think? Gram positive, gram negative?
Gram positive rod.
Very good. Gram positive rod. Look at this chain right here. There's 1, 2, 3, 4, 5, 6 cells all in a row, head to tail. This is that organism called Bacillus megaterium. It's just a really big rod. And I put this one in here to remind us again that you never know by the shape whether it's going to be gram positive or gram negative. In other words, you can have gram positive rods, you can have gram negative rods, you can have gram positive cocci, you can have gram negative cocci. You see all combinations. Now, the one thing that you do learn though with experience is that different species of bacteria have different sizes, even if they have the same shape. So Bacillus megaterium is a rod, but it's a huge rod. If you look at a rod like E. coli is much smaller. E. coli is also gram negative, not gram positive. And that's what you're looking at on this slide. This is an E. coli sample under a 40x lens. And then right next to it is the same sample under the 100x lens. So you can see the pink under 40x. And with some experience, you can see the rod shape under the 40x. But you can really see the details under the 100x. You can tell that these are rods. And they're pink. They're that pinky red, almost a fuchsia color when you go up under high power. This is E. coli, our old friend E. coli. And just like most bacteria, there are some E. coli that are pathogens. You've probably heard of the 0157 strain of E. coli. That's the really bad one that every year you hear about these outbreaks of E. coli. Last year or the year before, I guess it was, there was a terrible outbreak that involved romaine lettuce. And the CDC recommended that we stop buying romaine lettuce for a while because there was a lot of it that was contaminated with E. coli, and it was a bad strain of E. coli. But you also have E. coli living in your intestine, and you need that E. coli to be healthy. If you didn't have it living inside of you, you would not be your best, healthiest self. It's a different strain of the same organism. It's very similar, remember, when we talk about strains, it's very similar to when we talk about breeds of dogs. So you can have 2 dogs and they can both have the same scientific name. They're both called Canis familiaris. That's what a dog's scientific name is. But they can be different strains or breeds, right? You can have a Chihuahua and you can have a German Shepherd. The same is true of bacteria. All of this is E. coli or Escherichia coli. The genus name is E or Escherichia, and the species name is coli. Now, there are strains of E. coli, just like there are breeds of dogs. Some strains are harmless, and in fact, they're actually helpful to us. And other strains are pathogens. And if you're unlucky enough to get exposed to them, you can get very, very sick and even die from them. So again, it's important to remember that we can see bacteria under 40x and under 100x, but some bacteria are easier to see. So both of these are under 40x. These are those E. coli cells. It's kind of hard to tell that they're rods. You can see the pink. You know they're gram negative, but it can be a little hard to see that they're rod shaped. compare that to the Bacillus megaterium slide. This is also at 40x. The cells are clearly gram-positive, nice and purple, and they're clearly rod-shaped. This happens to just be a bigger cell. Bacillus megaterium is just a bigger organism, so it's easier to see under the 40x lens. Any questions? I'm saying a lot of names of bacteria today. Don't worry if you don't know how to spell something like Escherichia coli. You're going to have chances to see that word written out in this course. I try to use scientific names just so we get accustomed to hearing them. So again, during a lab period, if I'm saying it and it's not written out and you don't know how to spell it, don't worry. When we're moving forward in the course, you're going to get much more accustomed to seeing the names written out because they are horrible to spell. These scientific names are just horrible to spell. All right, let's talk for a minute about the limitations of this technique. All techniques, all diagnostic tests, it doesn't matter if you're in the microbiology lab or you're in some other kind of lab, they all have limitations to them. For example, gram staining is best done on a fresh culture. And when I say fresh, I mean less than 24 hours old. When you try to gram stain cells that are coming out of an old culture, say it's 2 days old or 3 days old, what'll happen is the cells will pick up the stain inconsistently. I don't know if you remember, but we looked at some cells the other day that had actually started to sporulate because they were in an old culture. The cells were running out of nutrients and they were stressed. And so they were starting to turn into spores. Now, not all bacteria can do that. Some bacteria, as they get old, they just start to die. And as a result, they don't take up stain very well. So the rule is you don't want to gram stain a culture that is older than 24 hours. You just won't get good results. Now, I have a couple of names written out right here. These are genus names. Remember, the first name is genus. The second name is species. So the genus Bacillus and the genus Clostridium are the two kinds of bacterial pathogens that can sporulate. So these are the organisms that cause diseases like anthrax, that's Bacillus. And there's a Clostridium called Clostridium tetani that causes a disease called tetanus. We get vaccinated against tetanus because it's such a serious disease. Well, Bacillus and Clostridium bacteria, the pathogenic kind of bacteria anyway, they can sporulate in culture. If they get stressed, if the culture starts to get old and those cells get stressed, they're going to start to sporulate. And sporulation will interfere with gram staining. So again, regardless of what genus and species you might be working with, you want to make sure that you have a fresh culture if you're going to do a gram stain. Something else to know about gram staining is right here. Gram staining, the stains themselves have expiration dates. When you buy one of those kits that has the 4 bottles in it, those kits come with an expiration date. Now, it's usually very generous. It's usually either 1 to 2 years that you can keep and use those stains. But remember, in some labs, they don't go through those bottles very quickly. And once you go past the expiration date, you really can't trust your stains anymore. So it's really important in a laboratory that we check expiration dates and we throw old stains away, because we don't want to get a false result when we're doing gram staining. So just like fresh cultures are best, fresh stains are best too. Take a look at this slide here. Take a really good look at it. I think you will agree some of these, well, first of all, they are cocci, they're round cells. We're only under 40x here, so they're not very big. But if you look around really carefully, some of these cells are purple and some of them are pink. Can you see that? Some of the cells, like up here, look kind of pink. Some of them are purple, some of them are light purple, some of them are dark purple. This is actually a pure culture. There's only one organism on this slide. It's a Staphylococcus. But this is a really hard gram stain to read because it's an old culture. This culture was 2 or 3 days old when this slide was made. And I think you'll agree, it's kind of hard to read it. It doesn't look like all nice dark purple or all nice bright pink. It's kind of in between. And this is what you get when you start to try to gram stain old cells. You just get an inconsistent staining pattern. Here's another one. These are rods, but gosh, are they gram positive? Are they gram negative? It's hard to tell, right? Some of them look really purple, but some of them look pink. Some of them are purple and pink. This happens to be a bacillus. This is one of the spore former types of bacteria, and it's an old culture. It's an old culture. So these cells are not healthy. They're running out of nutrients in the culture tube. They're accumulating a lot of waste materials in that culture tube. They're not in good shape. And so when you go to gram stain them, you get a really bad, poor result like this. Very hard to read. Now, let's say this was all you had to work with. Let's say all you had was this culture and you needed to figure out its gram status. What could you do? Well, you could mix up some fresh broth. You could take a few of these cells out of the culture, the old culture, and you could inoculate the fresh medium, the fresh culture medium, and you could grow a new fresh culture. But you would not want to draw any conclusions from this slide. Let's do a clinical application here. This happens to be a throat swab. So a patient came in complaining of a sore throat. The physician took a sterile cotton swab and swabbed it around in the back of that person's throat and sent it to the lab. Now you took that swab and you made a nice smear on a glass slide. You heat fixed it and you gram stained it. Now, this came out of somebody's throat. So this was not a nice pure culture that came out of a pure broth that you made in the lab. This is a clinical sample. And that's why the background of this slide doesn't look nice and clear. See these pink sort of amorphous areas? They don't look like cells. It just looks like a big smear of pink. That's just mucousy material that came in with the sample. What we're interested in are these dark purple things in the middle. What do you think?
Strep.
Do you think these are
Strep.
cocci or rods?
They're cocci.
Yeah.
It's strep throat.
good old fashioned streptococcus, good old fashioned strep throat. And again, one of the things we can see on this slide that's real classic for strep are these chains. See how the cells have lined up? You get these nice little chains forming on your gram stain slide. And that's kind of a giveaway for us that this is most likely strep. Good old strep throat. The only good thing about strep throat is that you can treat it. You can treat it with an antibiotic. Any more questions about that? Any questions about any of those slides that you've seen? Remember, the trick is after you make a slide, a gram stain slide, if I showed you, for example, a slide on a lab practical exam, and I said, what is the gram status of this organism? Remember, the first thing you have to figure out is it purple or is it pink? If it's purple, it's gram positive. If it's pink, it's gram negative. And then the other thing you have to figure out is what shape is it? Is it a coccus or is it a rod? Now, you'll notice that we didn't look at spirochetes today. We didn't look at that comma-shaped organism today. We just see primarily cocci rods in the lab. And those are the cells that we routinely gram stain. But it doesn't mean you can't get a gram status on other shapes. You just don't see them very often. All right. So coming up, remember that in an hour or two, once the recording of our meeting today has been all processed in Zoom, once it's sent to me, I'm going to post it on our playlist on YouTube. So if you need to go back in and you want to practice, you want to look at some of those slides again and practice determining gram status, you can do that. On Wednesday, when we get together, we're going to talk about aseptic technique. We're going to start, as we move into next week, we're going to start talking about some more testing that we can do in the laboratory. And so we're going to go through the basics of aseptic technique on Wednesday. The other thing we'll do on Wednesday, because it tends to be a shorter lab, we're going to talk about the semester assignment basically that you do in here for me. It's called the pathogen assignment or the pathogen project. So there's nothing to do for it. We're just going to talk about it on Wednesday. I'm going to tell you what you have to do for it and how I grade it, when it's due and things like that. So that'll be what's coming up for us on Wednesday. Don't forget, if you haven't already, and again, most of you have, but if you still need to take the lecture exam, you're going to want to get that done this evening. If something has happened, you need a little extra time to get it done, just let me know. And we can extend that due time for you. Otherwise, I will begin grading the lecture exams as soon as they all come in. And as soon as they're all graded, I'll post the grades. And typically that takes me, it takes me a few days to do that. So very good. Any questions? Any questions about anything that we've talked about? I'll leave you with a little bit of trivia, an important piece of trivia, I will say. There's one organism, a pathogen, that is kind of famous because it doesn't gram stain. If you remember, I said there is a gram status for this one kind of organism called gram indeterminate. You'll read that on laboratory reports. What it means when it's called indeterminate is that it's not purple or pink on gram staining. It just doesn't pick up the stains. And the type of organism, the genus of organism that has this feature is called Mycobacterium. And Mycobacterium, one particular species of it, causes the disease called tuberculosis. So whenever we suspect a person has TB, active TB, if we do a culture, if we get some sputum and we do a culture in the laboratory, whenever we try to gram stain it, we won't get a result. And that's a feature for that organism. That's something that is unique to that type of bacteria, Mycobacterium. The reason that tuberculosis organisms don't gram stain is because they literally secrete a wax substance. They secrete wax around themselves. It's very protective for them when they're out in the environment, including in your lungs and my lungs. It helps protect them from destruction, but it also makes it really hard to stain them in the laboratory because the wax keeps the stain out. So little piece of gram staining trivia for you there. All right, if everybody is set, we don't have any more questions, I'm going to let you go. Enjoy the rest of the evening. I will see you on Wednesday. And again, if you have anything come up before then, just message me and we'll see what we can do. Okay?
Thank
All
you.
right. Have a good evening,
Thank you.
everybody. We'll see you next
Bye.
time.